It's another concept to know that each of class 2 molecules have different alpha and beta chains encoded by different loci. Therefore, a fully heterozygous, this is a heterozygous because humans are outbreed population, can derive, as we see, there is 3 allelic variants from mother and 3 from father, but these 3, for example, from mother, encode both alpha and beta chains. So these are the same for the mother, these molecules, and this the same for the father. But combination of these can lead to this, as we see. So it could be, we can say there is a 12 allelic variants for MHC class 2 because of having alpha and beta chains. Actually, it was an interesting topic. Now we know how much is the capacity of MHC genes to express distinct peptides and how diversity is produced from heterozygous individual, especially from MHC class 2, and as we see MHC class 1. I will go on. Analysis of human HLA1, HLA class 1 genes from July 2012 reveals approximately 2013 A alleles, 2,605 B alleles, and 1,551 C alleles. And recall that all alleles do not encode because there is some non-classical MHC molecules. The number of HLA-DR beta chain genes may vary from 2 to 9 in different haplotypes with over 1,200 allelic forms. Actually, it's huge. But oppositely, DRA for the alpha chain is highly conserved, which encode only 2 proteins. So it is revealed by scientists that by calculating the numbers above of different genes or different allelic combinations, there is 1.7 billion different class 1 haplotypes in population. There is 1.7 different class 1 haplotypes in population, all human population. If we consider class 2, the number is even higher or even more staggering, with over 10 power 15 different class 2 combinations. And if we make the sum of both classes, it can reach to 1.7 plus 10 power 24 possible combinations. This is for a general population, not for an individual. Don't forget this. It's an important point because we said one individual, if for example in human population, a fully heterozygous individual can inherit 6 alleles for MHC class 1 and 12 alleles for MHC class 2. And this actually, it has some kind of, its advantages for the species because, for example, there could be some individuals in a species who are resistant, and if most of individuals in a species are susceptible, for example, they die, there is still some individuals which can survive because of having different MHC alleles which can display, for example, more important antigens to immune system and finally survive from the disease-causing agent. These are the number of allotypes, so these are the proteins encoded by these genes. You see, 1,448 different different structures, and so on. Actually, where this diversity is located, or the sequence divergence among alleles of MHC within a species is very high. This variation is not randomly distributed as we see in this peak. It is actually distributed mostly at the antigen interacting or peptide binding groove, which was formed by the beta sheets and at the floor and the alpha helices at the sides. So most of the polymorphism is located here. This is to maximize the diversity between MHC molecules of 17 amino acids previously shown to display significant polymorphism among HLA molecules. 15 were shown by X-ray class crystal graph analyzed to be in the peptide binding groove of this molecule. We are done with this, so now we know where it is located, and this is how it leads for recognition of different antigens. So what is the role of MHC? So what is the function of MHC? We said that they are expressed by different types of cells, and what is their function? For example, they have 5 basic functions. They can display self peptides or self antigens on class 1 molecules to demonstrate if whether the cell is healthy or not. And we will get back to this, how this is accomplished. Second one is to display foreign peptides in class 1 to show that the cell is infected and to engage with cytotoxic T cells. Third is to display self-peptide in class 1 and class 2 to test the development of T cells for autoreactivity. This is accomplished at the developmental time of T cells, for example, in thymus. Fourth one is to display self-peptides in class 1 and class 2 to maintain tolerance for self-proteins. And lastly is to display foreign peptides in class 2 to show the body if the body is infected and display them for T helper cells. So how these are done? By displaying different antigens or different kinds of MHC molecules to different cells. For example, how could we know the cell is healthy or not? For example, all of our body cells express MHC class 1, and the healthy cells should express the intracellular milieu. They should express antigens located or, processed, or they came from inside the cell to see. Natural killer cells will check, is it a normal protein or not? If it was not, for example, when the number of misfolded proteins increase, basically it means that there is a problem. About the second function, to display foreign peptides in class 1 to show the cell is infected. Yes, when the cell is infected, for example, a body cell, a somatic cell, for example, not only somatic, for example, when a body cell is infected, for example, with a virus, they display viral peptides concomitantly with self-peptides. So when a cytotoxic T cell engages with, directly leads to the killing of the cell. And this is to limit the replication of the virus. Also, some viruses have a strategy to evade immune system by dampening the expression of this MHC molecule, which in later we will talk about. Display self, the third one, display self to test for developing of autoreactivity during T cell development, Also, but it is special for T cell development in thymus. All body antigens are expressed by virtue of a gene called IRA in thymocytes. When T cell engages with this antigen and this MHC molecule at a very high affinity, I mean the antigen is self-peptide, they should immediately go to apoptosis and they should die, because if they survive, they will lead to autoreactivity. They will have reactions against our own self antigens. So this is danger. Uh, Another thing is to maintain tolerance. Actually, when T cell interacts with a low or intermediate affinity without expressing co-stimulation, this is from the cell which expresses the MHC molecule, this leads to unresponsiveness of the cells and is called tolerance or anergic T cells. Finally, how we can know if someone is infected? Actually, this is the function of antigen presenting cells, which are dendritic cells, macrophages, and B cells. What these simply do, they are located throughout the body in special places. For example, dendritic cells are rich in the dermis. So when there is a breach in this barrier, I mean skin, for example, some bacteria and some microbial antigens came in, these immediately engulf them and go to local lymph node to display them on MHC class 2, because it is from exogenous antigen, or it is an exogenous antigen processed by exogenous pathway. They are displayed on MHC class 2 to T helper cells to orchestrate an immune response. So this is how it is done. But know that not every time MHC expression lead to immune system activation. It is actually it differs according to the conditions. When or where or why MHC is expressed and how it is expressed, with co-stimulation, without co-stimulation, it is changes. So not every time leads to activation of immune system cells. What we now to talk about is that there is a different kind of antigen presenting cells. So these three are called professional antigen presenting cells. There is because they express MHC class 2 very well. There is some differences between them, which we will mention. For example, dendritic cells are viewed as the most effective antigen presenting cell because they constitutively express a high level of class 2 MHC molecules and have inherent co-stimulatory activity. They can activate naive T helper cells, but a macrophage, they should be activated by toll-like receptors, which are a part of innate immune system. Or, for example, by before they should, as I said, they should be activated, for example, by toll-like receptor before they express MHC class 2 or co-stimulatory molecules such as B7 ligand to activate T cells, T helper cells. And about the B cells, actually they can recognize antigen by B cell receptors, but as I told you, not all antigens can activate B cell, but they need T cell help. So before they should be activated by an antigen, by a cytokine, or a pathogen-associated molecular pattern before they express co-stimulatory molecule required for activating naive T cells. Even for its activation, it requires T cell help. There is also some non-professional antigen-presenting cells. These are located here. There is also some others. For example, mast cell, basophil, eosinophil, but they are not so significant. Before I complete this topic in this slide, we should know that MHC molecules are expressed on by different rate and different number in different kind of cells and different conditions. For example, lymphocytes are the cells which express the highest number of MHCs, and it is estimated to be approximately 5 plus 10 power 5. So it means that they can express this huge number, especially when they're activated. And there are some kind of other cells, for example, fibroblasts, neural cells, hepatocytes, they express the lowest number. And also it could be the reason that the tissue transplantation of liver could be sometimes are successful, or it is likelihood for rejection, it is less than drafting of other tissues. Okay, let's the conditions that can change the MHC expression pattern are these. For example, genetic. As we know, MHC molecules are expressed by, or it is encoded by some genes, or it could be multiple genes to involve in the process of transcription, process of assembly until it's expressed on the cell surface. And for example, defect in these transcriptional activators, this is class 2 transcriptional transactivator, or RFX, have been shown. Basically, these both have been shown to they are enhancers, or they promote the transcription of the gene. So when there is defect in both of these, it has been found in autoimmune deficiency. It's called lymphocyte Baer syndrome. So basically, lymphocytes lack the expression of class 2 MHC molecules, which lead to a very severe immunodeficiency. About viral interference, for example, as I told you, when cells become infected, they also display viral antigens to the surface so as to be recognized by CD8 T cell and killed. There are some viruses which interfere with this process to decrease the number of MHC, and so to evade the immune system and not be recognized, to finish the replication cycle. For example, CMV interferes with beta 2 microglobulin. It is shown before that in some experimental studies, when they kicked out the gene of beta 2 microglobulin in experimental animal, they saw that the MHC molecules is not expressed. When they returned back the gene of this molecule, it returned to its normal function, and it expressed. About adenovirus 12 caused decrease in this transcription of this transporter gene. Top 1 and top 2, what are these actually? About top 1 and top 2, both are involved in the process of transport of the molecule when it is transcribed, the MHC molecule, when it is transcribed, it is transportation throughout the cell, I mean, through from the cytoplasm into the endoplasmic reticulum, then to be assembled, it requires these type genes. So when it decreases, the virus interferes with this process simply. Again, the MHC molecules are not assembled and not expressed. Cytokine signaling and how this affects expression of MHC. Actually, among of these are interferon alpha, beta, and gamma, and the tumor necrosis factor alpha and lymphotoxin A. Each of these have shown to increase the expression of MHC class 1 molecule on cell. Typically, phagocytic cells that are involved in the innate immune response or locally infected cells are first to produce these MHC regulating cytokine. In particular, interferon alpha, which is expressed or is secreted from infected cells, for example, by virus or bacteria, and tumor necrosis factor secreted by antigen-presented cells after activation. This both upregulate class 1 MHC expression. So in later stages of the inflammatory process, also interferon gamma is secreted by T helper cells. This also contributes to increase in MHC molecular expression. Okay, what these factors do, binding these cytokines to their receptor induce an intracellular signaling. As you know, as a metabolic process, many things happen, and this leads to a signaling cascade to activate transcriptional factors. So what these factors basically do, they target the promoter of these genes and they increase the upregulation of the genes which encode MHC molecules. Also, not for interferon gamma not only increase the upregulation of MHC class 1, but also indirectly it leads to an increase in expression of CIITA, this one, which we talked about, class 2 transactivator, which subsequently leads to increase the expression of class 2 MHC. This happens especially on non-antigen presenting cells. As I told you, there is other cells which express MHC molecules, such as skin keratinocytes, intestinal epithelial cells, vascular epithelium, placental cell, and so on. Also, MHC expression on other cytokines, for example, MHC in only certain type of cell. For example, we haven't written here, interleukin-4 increased expression of class 2 MHC molecule only in resting B cells, turning them to more efficient antigen-presenting cells. Conversely, for example, interferon gamma, which previously we said it increases the upregulation of MHC class 1 and leads to indirectly for upregulation of MHC class 2 from non-antigen presenting cells. It is being revealed in studies that it leads to downregulation of MHC class 2 on B cells. So in different times and different cells, they have different effects. That's why what I point. So there is a last topic in this point, the third point, corticosteroids. So here we can see the importance of MHC molecules in the immune system. For example, most corticosteroids and prostaglandins can decrease expression of class 2 MHC molecules. Despite that, some of these are naturally occurring membrane permeable compounds bind to intracellular receptors and lead to a cascade which lead to downregulation of the class 2 MHC molecules and thereby downregulating or suppressing immune system. As you know, these are called also general immunosuppressors. I'm talking about the prostaglandins and corticosteroids. The last subject or the last topic, the last point in this slide is class 2 MHC alleles play critical role in immune responsiveness. And why? Because as you know, T helper cells are have the central role in immune system. For example, they can tell immune system, let's attack and let's activate, or let's begin to do an activity, or they can say no, there is no need for any activity. The antigen is safe and there is no need for any attack. When there is a problem here, the results are catastrophic. For example, it can lead to immune deficiency or it can lead to autoimmunity. There is two models. Scientists say that there is a model, determinant selection model. This is what we talked about, so that some individuals can express some peptides which could be more important for immune system to target them, and some other individuals could express some antigens which may be not important for expression. So this leads to the difference in immune response, for example, for the same antigen in different individuals in a species. There is also another model that holds in the repertoire model. It postulates that T cell bearing receptor that recognize certain foreign antigens which happen to closely resemble self antigens may be eliminated during T cell development. So this leads to holes in the immune system. And why? Leaving the organism without these cells for future response to antigens. For example, there could be some antigens which their shape be close from self-antigens. So in the absence of MHC molecule that can bind and present a particular peptide, or the absence of T-cell receptor that can recognize a given MHC molecule complex, both result in decreased immune responsiveness to a given foreign substance. This can account for the observed relationship between MHC haplotypes, which are derived between different individuals, and the response to particular exogenous antigen. About last two slides, T cells are restricted to recognize peptides presented in context of self-MHC, and how scientists reveal that. How they did? Simply what they did, in their experimental system, they derived macrophages from guinea pig, for example, from a certain kind of strain, for example, from strain 2, and incubated with an antigen. Okay, incubated with an antigen. After the antigen pulsed macrophage had processed the antigen and present on the surface, they were mixed with T cells from, for example, T cells from the same strain and different strains. Okay, okay. Now the macrophages are displaying the antigen, and they should activate T cells because they display antigen. But let's see what has happened. Actually, the T cells which were derived from the same strain, strain 2, could be activated by the macrophages derived from strain 2. But the T cells from strain 13 could not be activated. So this what led to this concept that T cells are restricted. So basically, they don't recognize the antigen alone, but there is also a role for MHC recognition. So this different haplotype cannot be recognized and activated by this macrophage which harbors MHC alleles from this strain 2. It could be any haplotype. There is another concept which is antigen processing before the displaying on MHC. Actually, it is a long and it also requires a special video on the antigen processing and displaying, but it will shortly talk about what the scientists did. They derived some antigen presenting cells. They let and they incubated with antigen. Later, they fixed with some kind of chemical, for example, paraformaldehyde, and they saw, despite that the cell has been stopped metabolically, but it can still activate T helper cells. And this is why, because before the fixation process, they have digested, processed, and displayed the antigen. But in this experiment, what they did, they fixed the cell before giving or incubation with antigen. So the cell is metabolically blocked and it can't do anything. It couldn't activate any T helper cells. And in another experiment, they fixed the cell before incubation with antigen, but they processed the antigen in vitro by exposing it to some proteases, and the antigen was digested. You see the difference between this and this one is so small, and it can accommodate to the MHC molecule. Also, the antigen presenting cells could activate T helper cells because the antigen was so small and was suitable to accommodate with the MHC molecule. So this what lead to that processing of antigen is a process which is required before the antigens could be displayed and recognized by T cells. So which later on, for example, if the co-stimulatory signal is expressed, it will lead to the activation of T helper cell, which later, for example, may lead to immune activation. This is a long process. God willing, we will talk about the antigen processing in a longer, on much details in next video. So subscribe my channel. Also, if you have any question or if you have any note, you can inform me at the comments. And thank you. We joined the end of our subject, and good luck.